Thanks for coming, everyone. We're starting off our next seminar series that we finished up our last one during the semester with Karolien giving an update on an overview of the Analytical Resources Core. And I will go ahead and turn it over, Karolien. Thanks.
Yeah, thanks, Corey, and welcome everyone. Thanks for taking the time out to come listen to kind of a high-level overview of the ARC. We have so much to offer. It's a little bit, it would take me a good day to talk to you about all our services, but I'm going to try and do that in 1 hour, tell you a little bit about who we are, what services we can provide you, and some of the latest developments here. If you saw my talk last year, I tend to just kind of point out what the newest additions are to the core since the previous year, things you may not know about us, and a couple slides on how you can get the most out of your time here working with us in the ARC. So yeah, I'm Karolien Denef. I'm the director of the Analytical Resources Core. And just a little bit about myself, just so you get to know who I am. Had a bit of an interesting journey to where I am now as the art director. So I started out in Belgium. That's where I'm from. Earned my undergraduate and PhD degrees in bioengineering from the University of Leuven. And for my PhD, I performed almost all of my research here at CSU at the Natural Resource Ecology Lab, studying soil carbon cycling and how especially agricultural practices and climate change impact those. And then from there, I went on to do a postdoc at the University of Ghent, where I actually got to manage a whole suite of really high-end and cool mass spectrometers to study ecosystem science at the molecular level. And that's when I really started liking the idea of helping researchers with advanced analytical tools that they need for their research. Then I took a 2-year break from academia and went to a global climate change consulting firm in Ghent, in Belgium, and I was kind of leading their new climate change consulting division. Then I came back to CSU in 2010 for a joint appointment as a research scientist at the NREL and as the managing director of a core there called EcoCore. And it was there that I really learned kind of the ropes of running an analytical core facility and really found my passion for making research happen through shared resources. So then in 2014, I joined the ARC, initially as a mass spec lab manager, and then in 2020 as the ARC director, and I've been with ARC ever since. So what drives me in my role is service. So really like helping researchers figure out the best way to get data they need. And I'm always looking for ways to improve our operations and think of innovation. So as the director of ARC, you might wonder what my job is. So I juggle a wide variety of roles to ensure the success of our core, and that can include mostly managing our day-to-day operations. I oversee our budget. I lead a really awesome, talented team of experts, very much involved in strategic planning. So I work on staffing, marketing, our instrumentation plans, and making sure that financially we stay sustainable. I do a lot of fundraising, mostly through writing federal grant proposals for new equipment, but also sometimes internally to secure some internal investments. And I handle lots of other jobs like customer relations, safety, compliance, our educational program, so that we can continue to train our users. So I wear many hats, and mostly in support of our 4 pillars. So those pillars are personnel. So I have to make sure that we have the right expertise and technical support in-house, our space and physical assets. So making sure that we keep cutting edge technology in-house and that we continue to stay relevant. Investment, it's really hard to live off of user income alone. That doesn't really sustain us. So we constantly need to look for support from grants and colleges and the VPR. And evaluation. So it's important in my role to stay aware of what our users need through surveys. We have an advisory board and through various feedback mechanisms. So it's a big job, but I really love the challenge of making ARC an important resource for all of you researchers here at CSU. So what is the ARC? Well, we're a CSU core, and that means that we are a shared research resource. So we provide shared resources to many research programs. We have a very broad impact, and we enable diverse research programs. We're also very much a place to learn for students to come and get experience. So education is a really big part of our mission. And we're also a team of really highly skilled people who are not only experts in their fields, but so they maintain the instruments and they collect the data, but they are also here to help you and support your research and give you the best possible experience here in the ARC. So that's what we really strive for. We're a unit in the OVPR. That's our administrative home, the Office of the Vice President for Research. But we're also one of many cores. So we're not the only core at CSU. There are lots of cores. I think in total is about 30 of them. So we're very well connected. So if you ever need something that we can't offer, we typically can put you in touch with the right person. So what does it mean? A shared research resource for ARC specifically. So we are a shared use analytical instrumentation facility that provides analytical capabilities and expertise that exceed the normal resources of individual research programs, and we make those technologies available as a shared resource. So the instrumentation that you'll find in the ARC are typically really high-end instruments that are typically too expensive or too sophisticated for individual PIs to acquire and maintain in their own research labs. And they're much more cost efficient if placed in a core that provides access to many users and that has the in-house expertise. So that's kind of how we operate. Researchers like yourself can come to us for chemical and structural analysis of small molecules. Think of metabolites or newly developed drugs by chemists. To look at macromolecules like proteins or polymers. To look at newly designed materials like coatings for medical devices or semiconductors or solar cells, or to take a look at the chemical composition or the morphology or the structure of functional materials through a whole suite of analytical technologies that we have available. We are part service facility where our staff runs your samples and we can deliver you with process data. But we're also part training facility where we can train you to operate some of our instruments, not all of them, yourself if you are deemed proficient. So we do have some strict requirements to make sure that you can independently run instruments safely and properly. Lots of instruments here. So I believe we are now at around 40 pieces of equipment, most of them pretty sophisticated, ranging from high field NMRs to benchtop spectrometers like FTIR and fluorimeter for chemical analysis. We have a DLS for nanoparticle size measurements, EPR for spin radical measurements. We have magnetic physical property measurement systems, a whole suite of mass spectrometers, depending on the types of chemistry that you want to look at. So either for untargeted workflows, you can use an LC-QTOF for targeted quantitative workflows. LC triple quads are typically used. MOLY for macromolecules, GCMS for volatiles, Orbitrap for proteins, ICP-MS for individual elements or trace metals. So we always have a lot of mass specs because every molecule requires a different mass spec technique. We have instruments for surface analysis, like our X-ray photoelectron spectrometer for looking at the chemistry of surfaces, say of thin films, semiconductors, profilometer for surface roughness. We have X-ray diffractometers to look at crystallography, phase analysis of inorganic samples, a powder XRD, a thin film XRD, and a single crystal XRD. And then finally, our electron microscopy lab has 2 SEMs, scanning electron microscopes, and a transmission electron microscope for ultra high resolution imaging and microanalysis. So those instruments and our services are structured in 3 centers in ARC. In ARC MMA, which stands for Materials and Molecular Analysis, That's where you can find all our expertise and instrumentation to do molecular structural elucidation, chemical and elemental analysis, EPR for spin radical characterization, thermal, magnetic, electrical properties of materials, the whole gamut. And you can do macromolecular characterization as well as X-ray crystallography and diffraction. Our ISS Center is Imaging and Surface Science. That's where you can find the expertise around atomic level imaging of materials, structure, morphology, elemental mapping of surfaces, all kinds of ways to analyze the properties and chemistry of material surfaces. We also have lots of sample prep prior to electron microscopy to prep hard and soft materials as well as biological samples for SEM and TEM. And then in our ARC Biocenter, which stands for Bioanalysis and Omics, that's where you can find instrumentation and expertise for both quantitative and qualitative metabolomics, proteomics, metaproteomics, bacterial identification, and ionomics, which is more the elemental composition of organisms. So I'm not going to go in detail on all these different services. Just know that we, each of these centers has a team of experts that work very closely together and you can reach out to for more information. So this is kind of our MMA team with myself, Indrani, in magnetics and materials analysis and spectroscopy. Ethan is responsible for XRD. Michelle is responsible for all our NMR services. Claudia oversees our self-use mass spectrometry. And Paul helps out in that area, and he's an expert in GCMS. Then we have our Biocenter with Corey as director, Gustavo and Dora supporting proteomics, Jackie in ionomics. Claudia is mass spec and NMR-based metabolomics. We've got links in targeted metabolomics, Nathan supporting untargeted metabolomics, and Grace helps out in both metabolomics divisions on the sample prep site. And then in imaging and surface science, we have Roy in charge of our TEM and Rebecca doing handling all our SEM services as well as surface analysis. So very strong collaborative team. People work cross center, sometimes get projects where we need skills in different areas because certain projects need multiple analytical approaches. So we work very closely together. So yeah, any of these people can help you out with more specific questions about technologies and services. They all also presented really nice overviews of their individual labs and services last year in our seminar series last year. All of those seminars are recorded. You can find them in our seminar library on YouTube. So definitely take a look there if you want to learn more about XRD or NMR. There's some really good overview seminars available there, and likely we'll repeat them throughout the coming years again, but definitely take a look. So the ARC serves a large and extremely diverse user group, mostly at CSU, but we also serve people outside of CSU. Past year, we've had over 900 individual service and training requests. That's quite a bit. We serve over 260 research programs at CSU, over 118 principal investigators from 34 departments or centers or units across 10 colleges. We also served 42 external for-profit industries and 17 nonprofit companies. So this map kind of shows you the dollar value of our services across the US and shows that institutions and companies all over the United States come to us for analytical services. So part of our mission is to enable STEM research and development programs that we nurture the early stages of experimentation and discovery at CSU. So in the next few slides, I'm going to show you a couple examples of research that were parts of their analytical work was done in the ARC and have led to a number of publications. So just to give you kind of a feeling of the breadth of research that we support. This first example is from J.B. Nielsen's group in the Department of Chemistry. They work on development of new materials for all kinds of different applications, including photovoltaics. So in this particular study, they were developing new solar cell materials that have high power conversion efficiencies. So some of these materials are unfortunately not very stable, and they undergo phase transitions to a different crystalline structure when it's triggered by water or light at room temperatures. And so it's very important to look at stabilization of those different materials, especially for solar cell applications. And that can be done through something called ion substitutions. And in this research project, the Nielsen group used solid state chemistry to integrate thiocyanate ions into the crystal structures of these new solar cell material candidates. And they used our single crystal XRD in the ARC to look at the detailed crystal structure of these materials. And their findings provided new insight that seemed to link observed crystal lattice defects to the stability properties of the solar cell material candidates. So you can do a lot with single crystal XRD to look at defects of materials, to understand the physical properties and the behavior of these new materials. The second study here is from Debbie Kranz's group, also a professor in chemistry. She's a synthetic organic chemist, and her group, her students work a lot around new drug development. In this particular case, they look at vanadium. Vanadium is a well-known transition metal that has anti-cancer properties. And in this case, they are looking at these compounds as potential drug candidates for treating brain tumors, or also called gliomas, by intratumoral injections. They would inject them straight into the tumor. And in this project, they used the ARC's NMR, so nuclear magnetic resonance, as well as EPR to characterize the new vanadium complexes that they synthesize in their lab. And specifically, they studied the effects of the chemical structure of these complexes on cellular uptake, cytotoxicity, redox chemistry, all very important factors for the efficacy of the drug. So powerful techniques, EPR and NMR, to really look at the chemical structure of new molecules developed for drug development. And these next two examples are from engineering. One is from Katul Popat's group. He's no longer at CSU, but this was done last year when they were still here. He was a professor of mechanical engineering, and they really work a lot around biomedical device engineering. So in this particular case, their study looked at surface modifications of titanium orthopedic implants that are used mostly in hip and knee replacements. And those surface modifications were important to reduce something that happens a lot on these medical devices, that is biofilm formation, which can lead to infections post-operation. And so they do that by modifying the topography and creating specific surfaces that decrease the adhesion of bacteria, and also help with integration into the bone. And in that study, they fabricated copper-modified titanium nanotube surfaces, and they used our SEM, scanning electron microscope, to look at surface morphology. They used something called energy dispersive spectroscopy and X-ray photoelectron spectroscopy to look at the chemistry of the surface, to see, how that copper modification was really developed on that surface. And then they used XRD to look at the crystallinity of those samples. So in these images, those are all SEM images that show you some of the bacterial formation on those surfaces or not. This particular case here on the right is from the Vulcans group in mechanical engineering. This group developed a very small wearable monitor called the AirPen, and that is a monitor that people can wear in a backpack to quantify exposure to fine particulate matter and volatile organic compounds that are toxic and that can affect a person's health and cause severe diseases. So things like benzene or toluene, xylenes, all pretty harmful. So the device contains a number of sensors, but also a sorbent tube that can collect the VOC. So you can see those tubes right here. They sit into this little wearable portable device. The VOCs get absorbed to that during the exposure period, and then those tubes can be brought to the ARC and analyzed on our GCMS to look at the chemical composition and to evaluate the exposure of the individual to all these harmful compounds. And then finally, one more example, more on the biological side. This is a research project of the Nachapa and Preni groups. It's a collaboration between faculty in Ag Bio and Horticulture and Landscape Architecture. They were working together on pest biology and management. In particular, on this research, this was looking at the management of the hemp russet mite, and that is one of the more serious pests for the hemp plant. And they use targeted quantitative metabolomic assays using our triple quad mass spec together with the expertise of LinkedIn to quantify a suite of cannabinoids following sulfur treatment of the mites as a management strategy. And what they found was that when mites were suppressed by sulfur, the hemp produced a lot more higher levels of cannabinoids. So triple quad mass spec really good for targeted quantitative small molecule work. So those are just a few examples of some of the research that happens here and the variety of analytical tools that people need for their research. Other things that we offer are training and education. So lots of people come here to learn. That can be done either through hands-on user training, where the ARC basically trains individual students on how to operate the instruments and collect data themselves. So on average, we receive about 40 new training requests on a variety of instrumentation. So it's quite a lot. We spend a lot of time on training. We also support class projects. So if you ever have a class where you want to do something analytically on instrumentation here in ARC, we're happy to support that. We have developed, thanks to COVID, kind of a virtual training program where we can give remote access to students and learn in classrooms remotely. Some of our vendors do demo days on new instruments that we are considering purchasing, and they want to place it maybe for a month in the lab to gauge some interest from faculty and students. We do some K through 12 education as well. One example is we get an annual visit by Lincoln Middle School here in town, and those students participate in the AVID program, and they come here for a couple hours to learn about chemistry and instrumentation, so it's fun. We offer short courses throughout the year, and our staff also occasionally does guest lectures for classes. So reach out if that's something that you're interested in. We'd be happy to support that. We also offer some professional training and workforce development. The ARC is a place where students can come and develop some professional development. That's done through our ARC Fellows program. So we hire frequently undergraduate students to help in the facility with sample prep, maintenance, running samples for people and delivering them results. So those can be hired through student hourly positions, work studies, but we also get student interns through official classes. We have postdocs that come and work here, kind of have a training program developed through an NSF MRI grant where one of the postdocs was paid by the grant to develop new methods and applications, which is always quite a lot of work when we acquire a new technology, the upfront time to develop methods and applications for new users. So usually we hire postdocs for that. And then GRAs are occasionally hired in the facility when we have kind of a temporary staffing shortage. When someone leaves and we need to fill a gap before we hire a permanent staff member, we typically ask for support from graduate students. So it's always been a really nice mechanism to keep the facility running. And faculty seem to be very supportive of that mechanism to help us out so that their research can progress. So like I said, we're one of many CSU cores. There's about 30 of them. If you want more information on that, you can go to this website. They're located mostly under different departments all across campus. The ARC is a little bit unique in that we are administered under the Vice President for Research. The only other core like us that is located under the VPR is Lab Animal Resources. So most other cores are typically administered under academic colleges and departments, but you can find out more information on this website. And yeah, the next few slides, I'll provide you with some useful tools and resources that can help you make the most out of the ARC in terms of how to find more information about us and our services and equipment, how to engage with us, where to request services and training, how to navigate iLab. It's a software tool we use for requesting services, for billing, for invoicing, and how you can just stay informed about the happenings here. So the first part is our website. So take a look at what is listed there. There's kind of a quick intro movie there. If someone ever asks you about the ARC and you just want to share some basic information about us, that's a good place to start. You can find an overview of all our services. We have a searchable table of all our equipment. You can click a button to request a service or training that will guide you to iLab. You can request a tour. Let's say you have a visiting graduate student or a faculty that's being recruited. We're always happy to give tours and show them the whole ARC if that's something that could help with that. We have, for people writing grants and need facilities overviews, we have a standard document with all our equipment and facilities described, and that's available on our website. You can find our rates there and all kinds of contact information. The second place is our user team. So we have a Microsoft Teams user team that currently has over 572 members. That's really kind of our internal communication platform where users get onboarded. They can find all our safety and training materials there. We have individual chat groups per instrument, per lab. So where basically the lab manager will communicate with users around instrument issues or questions about projects. So it all happens within Teams. And once you get onboarded, you'll become part of that. We also have an instrument status dashboard in our user team where you can see the status of our instruments, if an instrument is down, what the issue is. Sometimes we post alternatives. If it's going to be down for a few months, we typically help you out by connecting you with people at Boulder or in Anschutz or at Mines so that you at least are not stuck waiting for data for several months. Like I said, we use a software called iLab. It's just a web-based platform where all service requests and training requests are placed. It's also a place where all our instrument calendars live. So if you become a trained user, you can sign up for instrument time on a calendar that lives in iLab. So you'll probably create an account there if you're going to become a user. And then from there on, it's pretty easy to navigate it. We've got lots of SOPs around it. And we have a wonderful business manager, Kathy Griffin, who is a guru when it comes to iLab. So if you ever have issues with iLab, you just send her an email and chances are she'll quickly get back to you with a solution. We also have an email listserv, so if you haven't done so already, you should join it because that's where we post updates. We announce seminars and workshops. We have a monthly newsletter that features a student's work where they use the ARC, so it's always kind of fun to read what your fellow students at CSU are using this core facility for. There are updates about new people, new staff, new developments in the facility. Sometimes we post open positions if that's ever of interest to you, and information about new instrumentation and things that we might sunset. Like I said, we have a YouTube channel with lots of seminars, seminar recordings. You can also find some of our instrument training modules there. Some of them were recorded. Again, because of COVID, we had to kind of move to a virtual place to train people, and all our safety training modules are posted there as well. Or you can come to the facility and grab a few flyers. We have flyers for individual labs, for new instruments. So come and visit us and don't be shy to grab some materials. Every first Friday of the month, we offer tours. So if you're interested in seeing the entire ARC, not just the one lab that you've maybe worked with or that you're going to do services with in the coming months, we're happy to give you a full overview of all the different spaces and instruments that are here, introduce you to the people who manage them. So that is every Friday, and it starts in chemistry and it starts at 12 p.m. It's the first Friday of the month. So keep that in mind and sign up. Where can you find us? We are located in a number of buildings, all luckily very close to each other. So most of our self-service materials and molecular instrumentation and experts are all located in chemistry. So that's where you're going to find our NMRs, XRD, our materials analysis, instrumentation, spectroscopy, surface analysis, EPR, and self-use mass spec. Our chemistry research building over here, that houses 2 NMRs, soon 3, and our single crystal XRD. And then in microbiology, you'll find our omics mass spec facility and the life sciences stockroom. And then in Yates, you will find our electron microscopy lab. And then a few things you may not know about ARC. So I just gonna have a few slides, just tell you a few things that might be less known to you all. ARC participates in the My Green Lab program as part of our commitment to reduce our environmental footprint here at CSU. And some of the initiatives that we implemented last year resulted in a jump from bronze to gold certification. So we're pretty proud of that. Our goal is to continue until we reach green certification. So you might see some information about how you all as users can help with that. So things that we've implemented are things around our freezers, creating more awareness, greener procurement. So a whole range of initiatives that we're trying to do a little bit to reduce our footprint here on campus. You may not know, we have a life sciences stockroom that where you can come and buy enzymes or any reagents for molecular biology, kits. They're all available there. They can be special ordered if they're not available in stock. And Chrissy basically makes your life very easy because she does all the ordering, the processing, and billing. So it's a very convenient place for you to shop for biological reagents. So go visit her in microbiology or reach out to her via email. We also do every year an electron microscopy imaging contest, and the winners get a nice plaque of their image. So that could be TEM or SEM. And we have a big poster that hangs outside the hall that features some of the submissions. It's always fun. These are always some really cool images that are taken with our instruments. So stay tuned for that if you are into the imaging area. Something we just started offering are office hours. So a few of our staff are piloting this to see if it would help with students who need some extra help around development of instrument methods or data processing, data interpretation. So there's going to be some fixed times that staff will be available to help answer questions, help you with your research. So you can just book a slot on those times that are offered and drop in. So we're piloting this right now, but the plan is to expand this across all of ARC and have all ARC experts assist with this and help out. Maybe something else that you may not know is how we acquire instruments. Most of our instrumentation is funded through federal grants, so we do write quite a lot of proposals to get money to buy, high-end new sophisticated equipment. CSU is also very good at helping us out from time to time when we do need to replace aging equipment that are used by a lot of researchers. So that's kind of our second bucket. And then we do acquire sometimes instrumentation through leases that we then pay off through user fees where we have enough usage to make that doable. And then some is coming from foundation money and very little bit through donations. A couple of new announcements about new instrumentation. So last year we got funding for a new advanced SEM that is up and running. That was funded by the OVPR, and that was really to replace and upgrade a 23-year-old SEM, large user base, 46 PIs from 13 departments. So we had a really nice case for that. The main advances of this instrument compared to our old one is that it offers much higher resolution. It provides variable pressure, has a number of detectors ranging from EDS for elemental composition mapping to EBSD to inform you about the crystal structure and crystal orientation of material surfaces. New is that it will have a cathodoluminescence detector, which is really a good tool to look at trace element impurities and crystal defects. And that's very important, especially in geological research. It will have e-beam lithography. That's something that you can use, basically use a beam of electrons to pattern features down to sub 10 nanometer on substrates. And you will also have an air-free sample transfer, which is critical, especially I'd say it's very useful for battery research where samples are really sensitive to air and water exposure. So you can transfer your sample air-free into the SEM. So training is happening. If you're interested, you just contact Rebecca and she will connect with you to arrange for training. We're also getting 2 new NMRs. So that's very exciting. One of them is a 600 megahertz NMR that will come with solid state and liquid capabilities. That was funded by an NSF MRI grant. And very exciting because we haven't had solid state capabilities here at CSU for a very long time. So now people can actually perform chemical analysis by NMR on intact samples that you don't have to extract anything out of anymore to get it in the liquid phase. So think of soil samples, polymers, materials that were previously we were not able to analyze by NMR. So very exciting. That instrument will be located in the chemistry basement here in room C3E. And we're also getting a 400 megahertz NMR that will be placed in ChemR. This was funded by NIH and was mostly in response to a capacity issue. So our current 400 megahertz routine NMRs are quite heavily used. I think we were reaching 500 hours a month. So high need for extra capacity. And this system will have a 60 sample case. So we'll be able to do much more high throughput NMR work. So that's exciting. 2 new NMRs. Proposal efforts ongoing. We are trying to get an XPS funded to upgrade our 24-year-old system with a much more advanced system for surface chemistry characterization. That is now the 3rd round that we're trying with NSF MRI. We will try again in the 2nd round to USDA NIFA for an upgrade of our LC triple quad to do more sensitive analysis work for small molecule and metabolite screening. We're trying to get an upgrade of our thin film and powder XRD through federal appropriations requests. So that's really used for people working in the material science to look at chemical composition, crystal structures. And then in the future, we will hopefully work on an NSF MRI to replace our single crystal XRD with a dual source copper moly system. So lots of proposal writing happening for new instrumentation in the coming years. Couple new service offerings that you may not be aware of that are exciting, especially because they're collaborative efforts between various centers and disciplines within ARC. So our mass spec and NMR teams have been working on enhancing our traditional mass spec based metabolomics capabilities with NMR. Mass spec is definitely still popular and more traditional approach for metabolomics, but NMR does offer some unique benefits. It's very quantitative, requires very little sample prep. Things like separation and derivatization are not necessarily needed anymore if you're running samples by NMR. So it's able to detect a much wider range of compounds, including some of those that are more challenging by LCMS. So we hope to, by combining both, we hope to provide a little bit more of a comprehensive analysis of metabolic changes in biological systems. So if you're interested in that, talk with Corey, Linkson, and Claudia and Michelle, and they can kind of steer you in the right direction. Our electron microscopy and XRD teams are working together to develop a method to analyze small crystals by electron diffraction. That would be done using our TEM and some crystallography data processing expertise that we have in-house. So the benefit there is that we may be able to offer a new way to look at crystal structures to those that are too small to analyze by XRD. And then finally, we're trying to set up a small molecule crystallization screening technique. This is kind of a collaborative effort between our XRD and proteomics teams where the proteomics robot is being used to look at crystal screening, basically to optimize crystallization processes for users who don't really routinely perform crystallization work, but they require single crystal XRD for their research. So that might be a way to help them optimize their crystallization methods. We offer TEM class. That's something that might be of interest to you if you're ever wanting to do TEM. There's 2 sessions, each are 4 weeks. The first session is really focused on imaging by transmission electron microscopy. The second one is a little bit more advanced, working on electron diffraction and all kinds of microanalysis techniques in addition to imaging. So you get 1 to 2 hours a week of group lectures, 2 hours a week of private hands-on learning, and at the end you will be proficient to run the TEM independently. Roy Geis is the person to contact for more information about that. If you did not know, we have an advisory board. So if you ever have suggestions or some concerns about ARC, you can always reach out. You can always reach out to me. But if you want to reach out to someone in your college that is on this advisory board, these are the people who represent the individual colleges here. And we meet with them quarterly. So it's a good way for us to learn a little bit more about the needs of the faculty and the users in the different colleges. You can always reach out to me for any inquiries, to connect you to the right expert, to get information about onboarding and getting started with ARC. If you need class support, come see me. If there are any complaints or concerns, my door is always open for that. If you need help with grant support, any information about even the cores in the region, if we don't have a certain instrument here, but others do, I'm happy to connect you. I have a lot of people I'm in contact with frequently throughout the northern Front Range, the Front Range here. And then just a couple reminders. First one is about safety. So we require that anytime you enter the ARC, you always wear your safety glasses. Undergraduates are never allowed to be in our lab spaces alone. There has to be someone in the lab, otherwise they will need to go grab someone to accompany them. In the labs where we have high field magnets, magnet safety training is required. If you bring a visitor into the ARC, we ask you that you notify us so we can make them aware about safety. And you must be trained by ARC to operate any of the instruments, even if it's just assisting an undergraduate student with sample loading. We do ask you that you follow, official training so that you don't cause any risk to yourself, but also to the instruments. Finally, when you use the ARC, we ask you to acknowledge us by our research resource ID. It's something that allows us to find papers of people who are publishing data that was collected in the ARC. It's always fun for us to know what that data is being used for and how it helps you research. So it's a fun way for us to stay in touch with the research. You can follow us on LinkedIn. And finally, here's just a QR code for joining our mailing list. And that is all I had. So I am happy to answer some questions in the time that's left. So, or you can post them in the chat.
We did have a few questions in the chat already, Karolien, just so you're aware.
Okay.
I've tried to answer them in the chat, but there was interest in the citations, how we track the papers. So I put information in there on our ID. And First Fridays, I put the link in there for the tour request as well.
Great.
Other questions?
And if you cite us by your RID, we may pick you to feature you in our bulletin. So if that's something that interests you, we do a quick little interview and then we will post it on our bulletin, but you also get to appear on LinkedIn. So it's a little bit of marketing for you too. Any other questions? And yeah, the link for our tours, if you just go to our website, one of the buttons on the main ARC website is request a tour. So you can click on that and sign up. Oh, I think Corey already sent that link. Okay, good. Any other questions?